Cell reproduction part 2, control of the cell cycle. If the cell cycle occurred without regulation, cells might go from one phase to the next before they were ready. So what controls the cell cycle? How does the cell know when to grow, to synthesize DNA, and divide? The cell cycle is controlled mainly by regulatory proteins. These proteins control the cycle by signaling the cell to either start or delay the next phase of the cycle and ensure that the cell completes the previous phase before moving on. So this is a great time to pause and think about proteins and how many roles that they have that are based mainly on their shape. So you can imagine that certain conditions in the body could change the shape of the proteins that control cell division. For instance, pH level or heat. Think about maybe, if the body has a fever. A fever in the body can change the shape of proteins. Not that it would be, in this case, changing the cell cycle. That would be pretty major. But again, shape and proteins go together, and their function is dictated by their shape. Regulatory proteins control the cell cycle at key checkpoints. There are a number of main checkpoints. For instance, the G1 checkpoint occurs just before entry into the S phase, a synthesis phase. This checkpoint makes the key decision of whether the cell is big enough to divide. If the cell is not big enough, it goes into the resting period G. Okay, so you remember that G1 is a synthesis period of molecules. So maybe the molecules needed to move on have not been synthesized sufficiently. The second checkpoint is DNA or S checkpoint. The S checkpoint determines if the DNA has been replicated properly. So if we have of course, the chromosomes that have been duplicated in S have all been duplicated properly. Everyone has now another side to it, so to speak. Then this cell then can go on to the next checkpoint, which is the mitosis checkpoint. This ensures that all the chromosomes are properly aligned before the cell is allowed to divide. So this is where, if you think back at what happens with the cell, the DNA molecule has been duplicated, but then those chromosomes need to be pulled apart at the centromere into opposite ends of this cell so that two daughter cells can be produced. If we have too many of these duplicated chromosomes, perhaps they didn't get pulled apart, that go over to one side of the cell, or maybe if that happens, then we have fewer chromosome or genetic material in the other end of the cell, for example. So these proteins are checking for that even distribution of the chromosomes so that the cell can go on to division. So regulation of the cell cycle involves processes crucial to the survival of a cell. You saw, the cell has to be large enough, have everything it needs to be a cell, including organelles and special molecules, etc., and also the proper kind and amount of DNA. So the cell cycle regulates what happens by the detection and the repair of damage to DNA, as well as the prevention of uncontrolled cell division. So uncontrolled cell division can be as deadly to an organism as no cell division, and its prevention is crucial to survival. So we have, if we had cells that went on to divide that had DNA damage, then that could be a problem for the viability of the organism, depending on where those cells are. And if there were too many of the cells that they kept dividing beyond the point where they were needed, that kind of piled up on each other, didn't have these checkpoints that that allowed a pause in the division of the cell, then we'd have a problem too. Cancer is a disease that occurs when the cell cycle is no longer regulated. This happens because a cell's DNA becomes damaged, resulting in mutations in the genes that regulate the cell cycle. Damage can occur due to exposure to hazards such as radiation or toxic chemicals. Cancerous cells generally divide much faster than normal cells. They may form a mass of abnormal cells called a tumor. The rapidly dividing cells take up nutrients and space that normal cells need. This can damage tissues and organs and eventually lead to death. When uncontrolled cell division happens in the bone marrow, abnormal and non-functional blood cells are produced because the division is happening before the cell is ready for division. In these types of cancer, there is not any evident tumor. Cyclins and kinases. The cell cycle is controlled by protein-controlled feedback processes. Two types of proteins involved in the control of the cell cycle are kinases and cyclins. Cyclins activate cyclin-dependent kinases by binding to them. Now, again, this is another shape change protein example. When a protein has another protein attached to it, it can change its shape and change its function. This is kind of like what we see in the cell membrane when we had the channel proteins changing shape when something attaches to them. For instance, the phosphate molecule from ATP will change the shape of a protein. Cyclins comprise a group of proteins that are rapidly produced at key stages in the cell cycle. Once activated by a cyclin, this cyclin-dependent kinase activate or inactivate other target molecules through phosphorylation. Through this precise regulation of proteins, the cell cycle advances. Okay, now we have this term phosphorylation. We had it before talking about the ATP cycle in which, for instance, a molecule of ADP, adenosine diphosphate, is attached to another phosphate. So we've added a phosphate to the ADP. We also say that we have phosphorylated the ADP. And with that phosphorylation, there is a shape change, which is going to trigger some kind of change, which in this case, it's the storage of energy. Kinases are enzymes that catalyze the transfer of a phosphate group from ATP to another molecule in the cell. They function as a control switch in many cellular functions, turning a function on or off and regulating other cellular processes. Many times they are involved in activating a cascade of reactions. So we have our kinases now transferring a phosphate group. They're a protein enzyme that facilitates the removal of a phosphate from ATP to attach it to another molecule in the cell. And that attachment of that phosphate group can turn a switch on or off by making a shape change happen. Maybe it's turning the switch on or off on another enzyme that now has a different shape, and now it cannot attach to the reactants that it needs to have a certain outcome. What makes a cell cancerous? Cancerous cells divide without any normal limits. These cancerous cells can invade and destroy tissues, and they may spread throughout the body through what's called metastasis. The process by which normal cells are transformed into cancer cells is known as carcinogenesis, oncogenesis, or tumorigenesis. Mutations often cause cancer. Nearly all cancers are caused by mutations in the DNA of abnormal cells due to the effects of carcinogens. These carcinogens may act as an environmental trigger, stimulating the onset of cancer in certain individuals and not others. Do all people who smoke get cancer? No. Can secondhand smoke increase a non-smoking person's chance of developing cancer? Yes. And it also can increase other chances of other diseases developing. So having cancer is a combination of things, an environmental trigger perhaps, maybe genetic predisposition, and maybe there might be an effect from the body and its ability to actually counteract the overactive division of cells. The immune system plays a role also in targeting cells that are abnormal and getting rid of them. Complex interactions. Complex interactions between carcinogens and an individual's genome may explain why only some people develop cancer after exposure to an environmental trigger and others do not. Do all cancers need an environmental trigger? No. Cancer-causing mutations may also result from errors incorporated into the DNA during replication, or they may be inherited. Inherited mutations are present in all the cells of the organism. So if there's a mutation that's inherited, this would have come from the egg or the sperm cell, and then the egg and the sperm cell then in combination form the first cell of a new life, and then as that zygote, which is the first cell of a new life, has cells that are dividing, it will carry that mutation throughout the entire developing embryo. Proto-oncogenes and tumor suppressor genes. Some types of cancer occur because mutations in genes there are mutations in genes that control the cell cycle. Cancer-causing mutations most often occur in two types of regulatory genes called proto-oncogenes and tumor suppressor genes. Proto-oncogenes are genes that normally help cells divide. When a proto-oncogene mutates to become an oncogene, it is continuously active, even when it's not supposed to be. So a proto-oncogene is a normal gene that promotes cell division, but when it's mutated, then we have a action that is accelerated. This is like a car's accelerator pedal being stuck at full throttle. The car keeps racing at top speed. In the case of a cell, the cell keeps dividing out of control, which can lead to cancer. Of course, we want to consider that there are other factors that might prevent that development. Tumor suppressor genes are genes that normally slow down or stop cell division. When a mutation occurs in a tumor suppressor gene, it can no longer control cell division. This is like a car without brakes. The car can't be slowed or stopped. In the case of a cell, the cell keeps dividing out of control, which can lead to cancer. So we have these, a tumor suppressor gene and a pro-cell division gene working together in balance. But if one or the other is mutated, then we have an acceleration in cell division. Types of oncogenes. Oncogenes might be growth factors. So these are genes that can cause cancer. Protein kinases. Again, remember, shape changes can affect cell division. GTPases, which are enzymes that promote the development of GDP, which is a sister molecule to ATP, and which would provide phosphates for phosphorylation if that and with the result of perhaps changing shape of proteins that are regulatory, or transcription factors could be also oncogenes. So transcription factors would be involved in the transcribing of the DNA recipe into the RNA molecule. So if there are repetitions or too much of this transcription, then too much of a protein gets made, and that protein may affect cell division in a regulatory manner. Growth factors can be naturally occurring substances, usually a protein or steroid, capable of stimulating cellular growth, proliferation, and differentiation. They're important for regulating a variety of cellular processes. Usually they must bind to an extracellular or intracellular receptor to initiate a cellular reaction. So we have these factors that are proteins that are going to bind to something and change a shape, and changing that shape could lead to accelerated cell division. It may take several mutations to cause cancer. Cells have developed several control mechanisms to overcome mutations in proto-oncogenes. Therefore, a cell needs multiple mutations to transform into a cancerous cell. A mutation in one proto-oncogene would not cause cancer, as the effects of the mutation would be masked by the normal control of the cell cycle through those checkpoints and the actions of tumor suppressor genes. Similarly, a mutation in one of the tumor suppressor gene would not cause cancer either, due to the presence of many backup genes that duplicate its functions. It's only when enough proto-oncogenes have mutated into oncogenes and enough tumor suppressor genes have been deactivated that the cancerous transformation can begin, as signals for cell growth overwhelm the signals for growth regulation, and the cell quickly spirals out of control. Many of these genes regulate the processes that prevent most damage to the genes themselves, but DNA damage may accumulate as one ages. Mutations that cause cancer. Usually oncogenes are dominant alleles. We haven't talked about inheritance really in this course yet. A dominant allele is one of a possible pair of alleles that exist in cells that will overshadow another allele that we call the recessive allele. So if an individual has one dominant allele in the pair, the result of having that dominant allele will overshadow whatever the recessive allele is doing. So these are called gain of function mutations. The action of the mutant allele gene product, usually resulting in an activated protein, again, here are proteins, are dominant to the gene product produced by the normal allele. So they're going to overtake what the normal allele would do. So it's a gain of function. It is adding a function in. Meanwhile, mutated tumor suppressors are generally recessive alleles, as they contain loss of function mutations. A proto-oncogene needs only a mutation in one copy to generate an oncogene, but a tumor suppressor gene needs a mutation in both copies to render both products defective. So in tumor suppressor genes, and we'll see this again when we talk about inheritance more, need to occur in pairs to have an effect on cell division. So they're losing a function, which is losing the function of suppressing growth, needs two, but to accelerate the growth beyond what's normal, the proto-oncogene just needs one copy of a mutated gene. There are instances, however, when one mutated allele of a tumor suppressor gene can render the other copy non-functional. These instances result in what is known as a dominant negative effect. Okay, well, boy, this is a complicated subject. We're only just touching on this sort of thing here. One of the points is how very complex the interactions are in cell division and human health. Mutations that cause cancer. So what we have here is a example of what can happen. If a mutation activates a tumor suppressor gene, then the cell growth is not regulated down and cells proliferate. So the mutation might inactivate a DNA repair gene, which means that the DNA that's defective will be allowed to proceed through cell division. There could be a mutation of a proto-oncogene, which is going to accelerate cell growth. And then we might have another mutation that activates tumor suppressor genes. So we have a combination here of a lack of proper functioning of a proto-oncogene, which is going to allow cell division, a mutation in the tumor suppressor gene, which would slow cell division down, and then we have a mistake or an error in a checkpoint, which allows the defective DNA to move through cell division. And all of those put together now could have the possibility of allowing a tumor to form.